Hello, everyone, and welcome to today's webinar. I'm Deanna McNeil, Assistant Science Editor for The Scientist, and I will be moderating our discussion. Today, Mike Tayeb will discuss best practices for working with saliva-derived DNA samples from pre-analytical processing to result optimization. And Masita Arip will discuss sample type insights from stem cell transplantation, maximizing HLA-Match accuracy using saliva-derived DNA. Before we begin, I'd like to take a moment to thank today's webinar sponsor, DNA Genotek. We like our webinars to be interactive. We encourage you to send us your questions or comments at any point, and our speakers will address these during the Q&A session following the presentations. To ask a question, simply click on the Q&A tab and type your query into the question box located on the right side of your screen. This webinar, if you have minimized the Q&A panel, simply click on the blue chat button in the bottom right corner to bring it back. This webinar will be archived on the Scientist website, and we will send you the link via email within a couple of days. Please note that you will not be able to download the presentation slides. And with that, let me introduce our speakers. Mike Tayeb currently leads the product and application support team as a senior R&D manager at DNA Genotek. He is responsible for providing technical support to customers using products from DNA Genotek's molecular product portfolio, which is focused on providing sample collection solutions to the human genomics, microbiome, and infectious disease fields. He also leads multiple R&D projects to support product development and evaluate product performance. Masita Arip is a consultant pathologist for medical microbiology and immunology and the head of the Allergy and Immunology Research Center at the Institute for Medical Research, IMR, of National Institutes of Health of Ministry of Malaysia. She obtained her Master's of Pathology in Microbiology and Immunology at National University of Malaysia, UKM, and in her current role, she provides consultations to the Ministry of Health and non-MOH organizations for specific medical areas such as immunology, medical microbiology, vaccines, and laboratory quality management systems. She is also the Dean of the Diploma Medical Microbiology course at the IMR under the Southeast Asia Ministry of Education, Tropical Medicine. She studies allergy, autoimmunity, transplantation, immunology, tropical infectious diseases, and vaccines, and she established the Malaysian Stem Cell Registry in 2000, which is a database for healthy Malaysian volunteers who can donate blood stem cells to patients with blood disorders. Let's make sure that our slides are up and running. All right, take it away.
Hello, everyone, and thank you for joining us today. I'll start with a brief outline of what I'm going to talk about in the webinar. First, I'll cover pre-analytical sample processing, including best practices for saliva sample collection, options for DNA extraction, and some yield data from saliva samples extracted with different methodologies. I'll also provide some information on recommended methods for assessing saliva DNA quality. And I'll then pass the session over to Dr. Masita Arip, who will discuss her work with saliva-derived DNA at the Malaysian Stem Cell Registry. I'll start off with a brief description of saliva as a sample type. Approximately 70% of the human gDNA in saliva comes from white blood cells, with the rest coming from buccal epithelial cells. The figure on the right here shows a stained saliva smear under a microscope in which you can see several types of white blood cells listed as monocytes, lymphocytes, and segmented cells, as well as epithelial cells shown here. Saliva does also contain bacteria. However, when saliva is collected into a device that contains a stabilization reagent, such as that found in Oragene or OraCollect, bacterial growth is prevented and the amount of bacteria is minimized. So for example, if you compare Oragene to mouthwash or cyto brushes, the saliva collected into Oragene contains approximately 11% bacterial DNA on average, which is substantially lower than what you find in mouthwash and cyto brush samples. Mouthwash is about 66%, cyto brushes are about 88%. And the reason those are higher is due to their lack of stabilizing solution. For saliva sample collection, we recommend the use of Oragene and OraCollect saliva collection devices. Both of these devices are designed for untrained users to easily collect their own saliva samples without medical supervision. These devices, they're suitable for both in-clinic and at-home use. So OraCollect, which is shown on the left here, is a saliva swab device that uses an absorbent sponge to collect liquid saliva. After collection, the cap is unscrewed and inverted, placing the sponge into the stabilization solution for storage and transport. Oragene, which is shown on the right, is a direct spit saliva device that is designed to collect 1 to 2 milliliters of saliva. Users spit up to the fill-to line and then close this cap, which releases the stabilization solution into the sample. And the stabilization solution is designed to prevent DNA degradation and bacterial growth. A couple of nice features here is that the stabilization chemistry is sealed in the lid, so that there's no donor contact and it reduces errors. Additionally, there's a very clear fill-to line, which helps the donor collect an accurate sample volume. In order to maximize DNA yield and quality, there's a few simple rules that we recommend be followed when a sample is being collected. First of all, people that are collecting should refrain from eating, drinking, smoking, chewing gum, or brushing their teeth for 30 minutes prior to collecting a sample. These activities don't impact DNA quality, but they can significantly reduce DNA yield. Additionally, we recommend that the required volume of saliva be collected to maximize the DNA yield. And on the Oragene kits, we make that really easy with that clear fill-to line. So people collecting a sample know exactly how much saliva they should put into the kit. And lastly, the vast majority of users are able to collect a sample in less than 5 minutes. However, we recommend that they take no longer than 30 minutes. The reason that we do that is that we want to see the stabilization solution added promptly to the sample. People shouldn't take more than 30 minutes to collect. Beyond ensuring proper sample collection, we also recommend incubating collected samples at 50 degrees Celsius for 1 hour prior to performing the DNA extraction. This step can be conducted in the lab anytime after the samples are received and before DNA extraction. The reasons for this step, first of all, the incubation helps ensure that cell lysis is complete, and that leads to higher DNA yields. Second, it degrades proteins, which helps increase DNA quality. And lastly, it reduces sample viscosity, and that allows for easier sample pipetting, both manually and on various automated platforms. The incubation step is recommended. However, there are some workflows in which you may be able to skip this step. So for more information on this, please contact us. Our product and application support team will be able to help you determine if this step is necessary in your workflow. Our recommended method of DNA extraction for saliva samples collected using Origin and OraCollect is PrepIt L2P. And this is an extraction technology that's optimized for use with saliva collected into these devices, and it provides maximum DNA recovery. There's no columns or magnetic beads involved. It's a very simple and convenient workflow. And it does give reproducible results with very high molecular weight DNA and high quality DNA recovery. One of the other benefits is that the method is scalable based on the volume of saliva that you want to extract. So you can extract aliquots as small as 500 microliters up to 4 milliliters and anything in between. The extracted DNA is suitable for biobanking and also for a wide range of downstream applications, including PCR, microarrays, and DNA sequencing. And lastly, PrepIt L2P is a very cost-effective DNA extraction method. I'll show some data here that shows what results you can expect when you use PrepIt L2P to extract saliva. The mean DNA yield is 58 micrograms, and you can see that the majority of samples fall in the range of 10 to 100 micrograms of DNA. There are some samples that yield higher than 100 micrograms, but what's notable is that out of this set of 450 samples collected, there is very few. It's only 2 or 3 samples that fell below 10 micrograms total yield. So very high yielding extraction method. When you use PrepIt L2P with OraCollect samples, saliva collected with OraCollect, as shown on the right here, a 1 milliliter aliquot of samples yields between 1 and 8 micrograms of DNA, with the mean being about 4 micrograms. And this amount's sufficient for running a single assay, and there's enough left over for 1 or 2 retests if necessary. This chart also shows that the DNA yields from saliva collected in OraCollect is significantly higher than that of buccal swabs. The yields from buccal samples are often too low for some assays, especially when the increased bacterial DNA in a buccal swab sample is taken into account. And on the right-hand side here, we just have a figure that shows that OraCollect has a very soft, non-abrasive sponge tip for collection. And it's more suited for absorbing liquid saliva. When you compare that to abrasive buccal swabs, there's less bacteria that's collected because buccal swabs are used to scrape the cheek, which primarily collects epithelial cells and associated bacteria. For samples collected using Origin and OraCollect devices, if you're going to use those in a high throughput application, we offer another extraction method called PrepIt Q2A. And PrepIt Q2A is a rapid and scalable DNA isolation method. It's optimized for liquid-based removal of chemical inhibitors from Origin and OraCollect saliva samples. And what I mean by liquid-based is that there's no magnetic beads and no spin columns required for this methodology. It enables a direct-to-assay workflow for higher sample throughput. And in most cases, you don't need to quantify the DNA before using it in your assay. PrepIt Q2A is compatible with automated liquid handlers, such as the Hamilton Microlab Star, the TCAN Fluent, several others as well. And the DNA recovered is suitable for PCR, microarrays, and sequencing. Here I present some data from a couple of different assays. On this slide, we collected saliva using OraCollect here on the left-hand side and extracted it with PrepIt Q2A. And you can see that on Axiom microarrays here is the assay that we're using. You can see that DNA extracted with QIAGEN QIAamp in red and PrepIt Q2A in blue performs exactly the same on the microarrays. The call rates are both, with both methods, are sitting around 99%. And if you look across the bottom for the DNA input values, you can see that we included quite a wide range of DNA inputs onto the microarrays. So for PrepIt Q2A, ranging from about 50 to 300 or so nanograms input, and all of them ran successfully on the arrays. On the right-hand side, I have some metrics from sequencing of exome libraries prepared using library prep reagents from Twist Biosciences. And here we show that the performance of saliva DNA prepared with PrepIt Q2A is similar to gDNA extracted from a control cell line. So the first chart shows that we have a similar fold 80 base penalty score between the control and the saliva DNA. A very similar percent of reads were on target, and as well a similar number of duplicates in the sequencing data. And in this case, the saliva may have performed just slightly better than the control. In addition to PrepIt L2P and PrepIt Q2A, we have tested compatibility of Origin and OraCollect devices with other extraction technologies. There are a number of manual DNA extraction methods shown on the left-hand side of this slide that we have tested with, and they are compatible with many others as well. And then we have tested the samples on automated DNA extraction as well. So some smaller platforms like the QIAGEN QIAcube, for example, as well as larger instruments like the QIAsymphony and Hamilton Microlab Star and the TCAN Fluent. On this slide, I have some representative data from automated extraction of saliva collected using Origin. This first data set is extraction on the Thermo Kingfisher Flex using the MagMax magnetic bead technology. You can see the distribution of yields here across 48 different saliva samples. From a 500 microliter of saliva, the mean yield was approximately 4.3 micrograms. And we obtained A260-280 ratios around 1.93, with 98% of the samples greater than 1.8. So very good DNA quality as well. And you can see here on the lower panel that all of the samples had ratios above 1.8, except for one here, which fell at about 1.75. And just to wrap up the DNA extraction discussion, there are a few things that you want to consider when choosing DNA extraction methods to use with saliva samples. Primarily, you want a method that is optimized and best suited for saliva. So those would be prep at L2P and prep at Q2A, as I've discussed in this webinar. They're optimized for maximum DNA recovery. But the other considerations are, what are your throughput requirements? Prep at L2P is a manual process, so it's a little bit lower throughput than the automation, but an experienced lab technician can process about 48 samples in an hour and a half. Whereas prep at Q2A, it can be done manually, but it's also easily automatable. And on automation, you can expect a throughput of approximately 192 samples in 1 hour. So that's about 296 well plates. You also want to consider the input requirements of your assay. And more specifically, if you have an assay that requires a very high DNA input or requires very high DNA yields, you want to use an extraction method that is suitable for that. So prep at L2P gives you very high DNA yields and very high molecular weight. So it's suitable for very demanding applications like PCR-free based library prep and sequencing, or long read sequencing. And lastly, you want to consider the overall cost of your end-to-end workflow. And I'll talk very briefly about post-extraction DNA QC. A lot of the time, the common technique is to use absorbance at 260 nanometers for DNA quantification. And while this is quick and convenient and inexpensive, it can be inaccurate due to co-purified RNA, which can inflate your DNA yield as much as threefold. And that can negatively impact the performance of certain assays if it causes you to underload the assay. So for that reason, we recommend using DNA-specific fluorescence-based methods. In these cases, any RNA present doesn't contribute to the quantification. These are extremely accurate methods, and they provide better performance of downstream assays due to precise DNA loading. So some examples here would be Pico Green, Promega QuantaFleur, or Qubit. And lastly, if you're interested in looking at the fragment size of your recovered DNA, we recommend using a tape station where possible for most accurate fragment size assessment, but using an agarose gel is also acceptable. When you're extracting DNA from saliva using prep at L2P, you should expect to recover DNA with fragment sizes greater than 50 kilobases. And we show that on the figure on the right here. You can see 5 different samples extracted with prep at L2P, and you're getting DNA that's well above the top 48,000 kb marker. In most cases, these fragments are actually well above the maximum sizing capability of the tape station, which is 60 kilobases. It's really important to remember that extraction method does impact the size of the DNA recovered. So as shown in this gel, the lanes labeled SC are the exact same samples extracted using a silica spin column. And in that case, you can see the DNA fragment size is much smaller. It's approximately 15 kilobases. So if your application requires high molecular weight, such as long read sequencing, we recommend, again, using a method like prep at L2P. Lastly, I'll finish just with some highlights from a comparison of saliva collected with Oragene and OraCollect compared to the traditional buccal swab method. First, Oragene and OraCollect outperform buccal swabs in the stability, the DNA yield, and reduced bacterial contamination metrics. So if you look at this chart here, Oragene and OraCollect provide room temperature stability at ambient temperature for up to 5 years for Origene, for two or 3 months for OraCollect. Whereas with buccal swabs, you really only get a day or two of stability at ambient temperature. And buccal swabs often require drying in order to get stability. There's no cold chain required at all for Origene or OraCollect in order to get that stability. Origene and OraCollect, as mentioned previously, are bacteriostatic, and the amount of bacterial DNA is very low compared to a buccal swab, which could be as high as 80 or 90%. And the mean DNA yield from saliva collected in either of these two devices is somewhere between 5 and 50 times higher than the DNA yield from buccal swabs. So you have plenty of DNA for biobanking, for running multiple assays, for potentially having to run 1 or 2 retests. Whereas with a buccal swab, you may have problems running multiple assays or have to recollect if you have to retest a sample. So with that, I'll conclude here, but if you have any questions about saliva sample collection or use of saliva samples in your workflow or your assay, please reach out to us at info@dnagenotech.com. We have a very knowledgeable product and application support team that can assist you with just about every aspect of your project. And I'll now hand the session over to Dr. Masita Arip, and she will talk to you about her experiences with saliva-derived DNA.
Good day to all participants for this seminar. I'm Dr. Masita Arip. I will be presenting the stem cell transplantation, maximizing HLA-Match accuracy using saliva-derived DNA. Malaysian Stem Cell Registry, or MSCR, is established in December 2000. It is collaboration between Ministry of Health, Malaysia, which is Institute for Medical Research, and National Cancer Council for MACNA. Next. This picture showing the signing ceremony for second Memorandum of Understanding, MOU, MSCR between IMR and MACNA on February 10, 2020. So we are MSCR, one of the stem cell registry worldwide. What is MSCR? MSCR is a database or registry of people who are willing to donate their hematopoietic stem cells to patients in need. The objective of MSCR are to identify and collect data from potential donor volunteers, to give the chance to try and save someone's life from diseases that are treatable by stem cell transplantation, thirdly, to combat volunteers. Hematopoietic stem cells components, which are important because they can produce red blood cells for carrying out the oxygen in the blood, white blood cells to combat the infection, and platelet for coagulating factor. Hematopoietic stem cell transplant involve the replacement of patient's disease cells with those from a healthy matched donor. Hematopoietic stem cells are matched if both the patient and the donor have similar human leukocyte antigens or HLA. HLA are proteins that are found on the surface of most cells. So as you can see in this diagram, the patient white blood cell, if matched with the donor, so it can be transplanted. And if one of the HLA is not matched, the transplant cannot be done. So who needs a hematopoietic stem cell transplant? Mostly, most patients such as the leukemia, lymphoma, aplastic anemia, and thalassemia, they are the one who actually needs the hematopoietic stem cells transplant. Why would a cancer patient need a hematopoietic stem cell transplant? This is because chemotherapy and radiation therapy generally affect cells that divide rapidly. However, because bone marrow cells also divide frequently, high-dose treatments can severely damage or destroy the patient's bone marrow. So without healthy bone marrow, the patient is no longer able to make the blood cells. That's why the healthy transplanted stem cells can restore the bone marrow's ability to produce the blood cells that the patient needs. MSCR carries out the following activities. First, we do awareness, where we organize public and professional educational and awareness campaigns on the hematopoietic stem cells donation. Then during the awareness, whoever was to become the members, they will have to take the samples and we run the HLA typing for the members of the MSCR. And this registration and test will be maintained in database, and there will be a search within this database to find the match donor. Currently, up to December 2023, there are 39,105 members recruitment from MSCR. So who can register as a donor? First, it must be Malaysian citizen, aged between 18 to 50, and healthy potential blood donor. Next. After fulfill the criteria to become a member, there will be a briefing on MSCR on the role of blood stem cell, what is blood stem cells, and criteria to be in the MSCR registry. Then the people who are volunteers will fill in the MSCR registration form and take specimen for HLA test. So the sample recruitment will be either 10 ml of blood, 2 ml saliva, and saliva swab. The members can be recruited either by using the open booth, or the people who volunteer will walk into our institute, and we will also give a talk to the colleges or universities for MSCR campaign, and members also can enroll during using the online recruitment. Next. So these are the type of sample collection method, either blood, saliva, or saliva swab. So after we take the samples, the DNA needs to be extracted, and we will run the HLA test, and all this information will be kept in the MSCR database. I will talk a little bit about the test, about the human leukocyte antigen, HLA. This is a membrane-bound glycoprotein that binds processed antigen peptides and present them to T cells. There are two types of the HLA, which are the main HLA type. HLA class 1, comprised of A, B, and C, and HLA class 2, the RDQ and DP. And as you can see here in the diagram, the nomenclature for the HLA will be the HLA, and then there will be a locus, either A, B, C, the RDQ, DP, and then there will be the allele family. For example, in this case, 24, and the allele number 02. So HLA allele numbers quite huge. For example, the allele for HLA-A is 8,000 something. So as you can see here, there will be a quite polymorphism of the HLA alleles. Next. So each individual inherits sets of HLA genes from both father and mother, and the set of alleles inherited from each parent from a haplotype. So if you can see here in this diagram, if the child is number 1, if the child is patient, there will be child number 3 can donate the stem cells. But if the patient actually child number 2, they need to search to the unrelated one because there's no match in the siblings. So the chances of HLA match only 25% between sibling donors. There are many methods for HLA tests. The first method, which is the serotyping, is a very traditional way of doing HLA tests using the protein-based detection. However, they are not really accurate in terms of the HLA typing. The recent technology is a sequence-specific oligonucleotide hybridization, or SSO, where this method actually target the specific oligos to detect the HLA from the DNA patient or donor. However, it is not really specific. The gold standard for the HLA test is actually sequencing, either Sanger sequencing or NGS, next generation sequencing. So DNA-based HLA typing is easily extracted from any nucleated cells, and DNA also easily stored, allowing repeat sample testing when required, and based on the PCR application of target sequence in the HLA gene. So PCR primers and oligonucleotide probes can be designed and validated in-house or purchased commercially. These are the example of the HLA matching between the patient and donors. If you can see in the above, the green box, they will be all 100% same HLA alleles. As compared to the red one, there's a slightly different HLA alleles, which is DR1501, and for the patient, DR1502. So the red one is considered not matching. In Malaysia, we have National Stem Cell Coordinating Center, NSCCC, located in the National Blood Bank. So this NSCCC will become the coordinator between the Malaysian Stem Cell Registry, Blood Bank, and also if the transplant center or the hematologist need to treat the patient, they will search in the NSCCC. And this NSCC will search in this two database, MSCR and Blood Bank. And NSCC also will coordinate if there is a match from the MSCR to bring the patient to the MSCR donors to the collection center to take their blood stem cells. For full match donor workflow, they will undergo the confirmatory HLA typing, which must be matched between 10 antigen match, and there will be a briefing and counseling about the collection procedure. There will be a physical and blood investigation done. The donor will have to go to the hospital to take the blood stem cells. So in the collection center, the hematopoietic stem cell will be collected either the bone marrow aspiration or FRCs. For Malaysian Stem Cell Registry donors, they will be given a special card from the Ministry of Health, where there will be a free of charge of admission to first class ward and free of charge for any treatment in Ministry of Health hospitals and clinic. These are the example of the identification card for the donors from MOH. Currently, we already have 21 match unrelated donor from MSCR, started from 2010 until 2020-21. And if you can see here, the risk between patient and donor must be the same in order for them to donate their stem cells. Out of 21, 16 Malay donors and 5 Chinese donors. And out of these 21, one actually already donated, only one actually donated the bone marrow. The remaining donate using the FRCs method. I would like to share the successful story of a patient treated with hematopoietic stem cells from the MSCR MAT. This actually happened in 2013, where the donor is female and Malay. She actually donated the bone marrow donation, and the patient actually having acute lymphoblastic leukemia. And as you can see now, patient actually survived. This picture in 2017 with his mother came to our office, and now patient already aged 24 years old and married. I would like also to share about the chronology of MSCR sample collection method. We started using blood to collect the sample for HLA typing, followed by with saliva and saliva swab. The limitation for the blood sample for HLA test, first, of course, is quite invasive. We need to prick the patient. Secondly, the donor compliance also quite challenge because we need to take the blood, and some of the volunteers actually quite phobia with the blood. Actually, they can easily fainted, and also if there is a large number of volunteers, it's quite difficult also for us to take the blood. And we need also to have medical officers or paramedics during the blood taking. And the process also very slow sometimes when we have a difficult pains. And we need also to have a special temperature to transport the blood in the DTA tube. And lastly, the blood must be processed immediately. The advantages, of course, the high DNA yield and low cost of DTA tubes. Because of that, we evaluate the using of saliva, and this evaluation we presented in the 10th annual scientific meeting of College of Pathologists in Langkawi in 2011. I will share about the findings of this evaluation next. So in this evaluation, we introduced about the self-collection of saliva using the origin from DNA Genotek to prevent the DNA degradation and pre-treatments of cells to prevent bacterial growth present in saliva prior to laboratory processing. And the aim of this study is to evaluate the quality and quantity of DNA using the origin saliva collection method and to determine its suitability for HLA typing. In terms of the methods, 14 individuals consented to deposit 2 ml of saliva into origin saliva collection tube. DNA was extracted using a standardized protocol provided by DNA Genotek for the origin, and the concentration of DNA extracts was measured. HLA typing was carried out using PCR SSP and PCR SSO. This diagram showing the PCR SSP as using the, as you can see, there is a band, and for the PCR SSO, there will be the analysis of the results. And HLA typing that was carried out from saliva was compared with DNA extracted blood. The results, the participants of these studies were 13 female and 1 male, and 12 out of 14 patients are Malay, and the remaining 2 are Indians. The mean DNA yield from saliva was 147 nanogram per microliter. It ranged from 9.4 to 580 nanogram per microliter. 8 HLA-A, 13 HLA-B, and 8 HLA-DR alleles were identified. In conclusion, we have demonstrated that saliva samples provide sufficient DNA for HLA typing by PCR SSP and PCR SSO methods with results comparable to the DNA from blood. We changed from the blood sample to the saliva sample for HLA test, where the volunteers will need to spit out 2 ml saliva, and there are many advantages. First, it is painless and non-invasive, and donor also prefer to use the saliva. Lower cost of personnel, sufficient DNA yield for assay, no need for medical officers or paramedics, faster sample collection, doesn't need special temperature for transport, and the saliva sample also can be kept at room temperature for up to 5 years. From saliva swab, we actually changed to saliva swab in 2016. The reason is because the saliva swab is more convenient compared to saliva in terms of lightweight, easy to handle, especially in bulk, more compact than in saliva. Sampling is easier as not everyone can produce saliva in high amounts, more cost effective, and less time consuming during sampling. And the advantages of saliva swab are again similar with the saliva, such as no need medical officers, faster sample collection, doesn't need special temperature for transport, can be kept at room temperature, and more cost effective compared with saliva kit. So these are the differences between the saliva and saliva swab in terms of the DNA range of probability for saliva between 1.7 to 1.93, and concentration between 500 microgram per ml to 200 microgram per ml. Saliva swab, the range of probability between 1.66 to 1.98, and concentration normally below than 100 microgram per ml. But all this is sufficient to run the HLA test. This diagram showing the usage of saliva swab per year in our institute, and it keeps increasing, and we estimated that we will use it more than 2,300 for this year. Okay, this video showing the campaign at Malaysian Police Training Center, Langkawi, Kedah. As you can see, we can ask them to do the saliva swab all together, and they are very excited also to use this saliva swab. I would like to take this opportunity to thank my team, MSCR team. All of them are really hardworking personnel, and they're working even during weekends. That's all from my presentation. If you want to know more about our MSCR, you can see our Facebook and our website. Thank you very much.
Thank you both so much for that presentation. As a reminder to our audience, feel free to submit your questions at any point using the Q&A tab on the right. We do have some questions already, so we can jump in to the Q&A right now. Let's start with one for you, Mike. We have a question from the audience asking about whether you have a suggestion for a low 260-230 ratio.
Yeah, so I think it's important to first mention that the 260-230 ratios from saliva will be variable. Sometimes they're high, sometimes they're low. What we've found is that there is no correlation between the performance of the DNA in a downstream application and the 260-230 ratio. So for that reason, we don't really recommend using the 260-230 ratio as a metric to evaluate whether the DNA will perform downstream or not. So if your 260-280 ratio is good and your yields or concentrations are good, we suggest that you proceed with the sample regardless of 260-230 ratio.
Perfect, thank you. We have a question for Dr. Masita. An audience member says, I understand that your study was held in 2011. Is there any difference in the Origene product used for the study and the current Origene products?
Thank you, Latifa, for the question. From our experience, using saliva and saliva swab, we are satisfied with both products. Both can, we can take the DNA from the saliva and saliva swab, and we can run the HLA typing either using the SSO or NGS. That's my answer for the question.
Perfect, thank you so much. Have another one for you, Mike. With the 50-degree incubation prior to extraction, does it require an addition of a protease K treatment?
Okay. No, so there's no need to add any protease or proteinase K to the sample prior to the incubation. So the stabilizing solution in both the Origene and the OraCollect sample or saliva collection kits, it contains all the reagents that is needed for protein degradation. So you just have to take the sample, put it in the 50-degree incubator, no need to add anything to it.
I see, thank you so much. Dr. Masita, do you expect any difference in the performance between saliva versus saliva swab for HLA testing?
At the moment, from what our experience in terms of the performance, I think both performance satisfactory, where we can achieve our target to run the HLA test using the DNA obtained from the both saliva swab and saliva. So there is no so much difference in terms of the DNA from both type of specimen collection.
Perfect, thank you. Let's see, Mike, and we have an audience member who says, thank you for the informative presentation. I'm wondering if there is a workflow for extracting cell-free DNA in saliva samples.
So for cell-free DNA with an Origene or OraCollect device, it's not really possible to get cell-free DNA from the device. The preservative in there is actually lytic. So it will release cellular DNA into the solution. So the separation of cfDNA and cellular DNA isn't really feasible. The reason that it's lytic is because that's one method by which we help stabilize the DNA. So yeah, unfortunately, you wouldn't be able to get cfDNA from those samples.
Okay, perfect, good to know. Dr. Masita, we have a question from the audience asking which extraction process you used for saliva versus saliva swab.
The answer actually we are using the similar protocol actually, using the reagent PrepIt L2P. That's the name of the reagent for extraction the saliva swab.
Perfect. Thanks so much. Let's see. A question for Mike. Are there, actually, there's quite a few questions for Mike. We can start with the first one. How does eating prior to saliva sample collection affect DNA yield?
That's a good question. So really what happens when someone eats is that the act of eating clears free saliva out of the mouth. So first of all, the person has less available saliva to collect at that particular moment. And when freshly generated saliva doesn't contain quite as many cells. So when you eat, you're really clearing away the cells from the mouth. And it takes about 30 minutes to regenerate, good cell content in the saliva. So that's the reason that we recommend you wait 30 minutes between eating and collecting a sample.
I see. And then in the same line of questions, this question asker is saying, we had difficulty collecting saliva from babies. To induce saliva, we used liquid glucose in the baby's mouth. Is this advisable, or do you have any other suggestions for this?
Yeah. Okay, so I have a couple of suggestions there. Using something like liquid glucose is acceptable. You just want to keep the amount of glucose used to be fairly small. It will induce saliva production. Also, something like a drop of lemon juice works. Something that's sour will induce salivation. But the other thing is that very young babies, so under about 9 months old, they don't produce much saliva to begin with. So for that reason, we recommend using the saliva swab-based kit, which is OraCollect, because it will collect some saliva, but it'll also collect epithelial cells. So that may work better for you in the case of infants.
Perfect. And then the third part of this question is, our sample collection site is far from the lab, requiring a 3-day journey. During transport, the temperature might reach 40 degrees Celsius. Is this acceptable? And what is the maximum recommended temperature for the samples?
Yeah, so these sample collection kits, once the sample's collected, it was designed to withstand temperatures that you would experience during transport. So the samples have been tested for stability up to 50 degrees Celsius. So they can withstand several days at least at 50 degrees Celsius. So during transport, it won't be a problem. You won't have temperature-induced degradation of samples.
Okay, fantastic. For Dr. Masita, how was patient compliance with Origin and the OraCollect kits? How did the patients like the kits, I guess?
Yeah, I think for both either saliva or saliva swab, it's easy to use, and it's not invasive. So that's why I think our MSCR volunteers like to use this as the sample collection as compared to blood sample. So same also with the patient in the hospital. If the advantage using the saliva swab or saliva sample, if there is a patient already transfused with recent blood transfusion, it's quite difficult to test for their HLA test. So we recommend them to use the saliva or saliva swab for the HLA test. This is from our experience.
Perfect. Thank you. I think we might have time for a couple more. Let's see. Mike, we have a question. It kind of follows up a bit about what you're speaking about temperature, but regarding saliva collection, once collected, how long can it withstand room temperature prior to extraction?
Yeah, so if you're collecting saliva, like direct spit saliva into an origin kit, the stability period is actually up to 5 years at room temperature. So it's a very long time at room temperature. And for AuraCollect, which is the saliva swab type device, you'll get at least 60 days of stability out of it. We have found that it usually lasts longer than 60 days, but our official claim is 60 days.
Perfect. Thank you. Dr. Masita, do you have data regarding the percentage of saliva swab saliva test needed to proceed with blood tests for HLA?
Yes, we have. At the moment, we notice that less than 3% of our DNA extracted from the saliva cannot run the test, so we need to take from the blood. Less than 3%.
Perfect. Thank you. Question for Mike. Are there any cons or negative effects beyond 1 hour of incubation?
No, there aren't any negative effects beyond 1 hour. In fact, we have tested incubation for much longer periods, like multiple days over the course of a long weekend, for example. And it's actually fine if you need to leave the samples in the incubator longer. What some people do is they collect a sample, or they put a sample in the incubator the night before that they want to extract it. They leave it there overnight, and then they extract in the morning, and that saves them, 1 hour the next morning waiting for an incubation. So it's perfectly fine to go longer, up to several days.
Okay, awesome. Thank you. I have a question for both of you. Do you have any experience with saliva swab and saliva collection in pediatric patients, specifically looking for high DNA yield? Dr. Masita, would you like to go first?
Yes, we do have experience for both saliva and saliva swab for pediatric patient when they are requesting the test for HLA in our diagnostic lab. And we satisfied with the performance of the DNA collected from the saliva or saliva swab.
And we've definitely used both saliva and the saliva swabs with pediatric patients, and it's easy to get enough DNA yield to do most downstream applications like sequencing or qPCR based assays. So there shouldn't be any problems with pediatric patients. We do have some assisted collection versions of Origin, which if the child is not able to spit directly into a kit, you can use a swab or a sponge to collect saliva. But other than that, the samples work fine for pediatric use.
Perfect. Thank you both. Another question for Mike. Can the kits be used for DNA methylation studies?
Oh, yeah, that's a good question. Definitely can be used for methylation studies. We have tested a couple of different methylation applications. One is on the methyl epic microarrays from Illumina. And we have also done whole genome bisulfite sequencing from saliva. Both of them worked very well. And there are multiple publications out there that if you look up origin or collect and methylation, you should be able to find them. Multiple publications have shown that it can be done successfully.
Perfect, thank you. I think maybe we'll have time for one more. This audience member says, "Hi, Mike, you mentioned that the buffer in the collection device has a lytic property. Is there any concern with this lysis property when combined with lysis buffer in a DNA extraction kit, such as a lysing sample, for too long or exposing a sample that is already lysed with stabilizing buffer?"
Okay. So as far as it being lytic, and is there any problem with it, being lysed for too long? No, there isn't. Really, once the cells are lysed and the DNA is exposed to the stabilizing solution, that's actually when the sample is most stable. Is when the DNA is released from the cells, and that's because the stabilizing solution, one of the things it does is it inactivates nucleases that are present in the sample. And then I think the second part of that question was, is there any compatibility issues with lysis buffers in extraction kits? We have never run into a situation where there's an incompatibility with an extraction kit. As far as everything that we've tested, all the commercial kits that we've tested, you can mix the lysis buffer in the extraction with the stabilizing solution in a saliva sample, and they're perfectly compatible with each other. So there shouldn't be any concerns there.
Okay, perfect. Thank you. And thank you both so much for that discussion. We really appreciate you joining us here today.
Thank you, everyone.
If anyone has further questions, please do consider reaching out to the speakers directly. Their contact is shown on the screen, as is a link to request samples of Origin, OraCollect, and/or PrepIt reagents. As a reminder, this webinar will be archived on the scientist website, and you will receive an email notifying you when the on-demand webinar is available. Please make sure to follow the scientist on social media for updates on future webinars. I'd like to thank everyone who took the time to join us today, and particularly those of you who shared your wonderful questions and comments. On behalf of the scientists, I'd also like to thank our speakers one more time, Mike Tayeb and Masita Arip, as well, of course, as our sponsor, DNA Genotek. Thank you so much, everyone, and goodbye.
Thank you. Bye.
Thank you.